All right, good afternoon, everybody. Welcome to Monday's Herpetology class and your first quiz of the semester, which is very exciting. Just real quick, here's what we got in store for us today. We're going to bounce back and cover amphibians again today. I know we jumped into reptile diversity a little bit last week, and our agenda today looks like this. We're going to take the quiz to get started, and I've got a short video to clear your head after you take the quiz. Bullfrog versus newt. Battle. We'll see. And then I want to correct an error that was Princeton pointed out to me from last lecture, and then talk a little bit about the Ologies podcast that you listened to today. I hope you learned some and enjoyed it. And then we are going to watch the some Life in Cold Blood, and we have kind of directed questions, and I'll probably pause it a lot and just talk about stuff, which will be really annoying, but you'll learn about herpetology, so it's a trade-off. We'll wrap things up and talk about upcoming assignments and everything like that. So first things first will be the quiz. Before I pass these out and we get started, what questions do you have for me? All right, the quiz is closed book, so please go ahead and put your stuff away. I'm going to pass this out. You have 20 minutes to take it. I'll set a timer once everybody gets theirs. I'll try to pass them out quickly. Actually, Princeton, can you help me pass these out? Seal to play. I'll just give you half. If you have any questions about anything anytime, just raise your hand and I will answer for you. Don't feel like you can't ask a question during the quiz, because that is a terrible feeling to have. Go ahead and get started. Yeah, go ahead. I just want to make sure. I always check to make sure you've answered all the questions when I collect it. That's it, Sam. Just make sure you get everything answered. Yes, that's it. Just make sure you get everything answered here. It's good. Thank you. I guess I didn't need to be on this. Oh well, at this point I'll just leave the recording. Let's fast forward through that.
Did you link that?
No, I have. I found like I don't know, some educational service or something made this like it's a whole PowerPoint with all these different timers in different formats with different minute amounts on it. I use them all the time in class.
Wow.
You got these? Thank you. Yeah, they're super handy.
This is all crazy.
Okay. I think that's it. All right, I think that's everybody's quiz in. Is that right? All right, great. Good job, everyone. I will grade these as quickly as I can and get those grades back to you. I know that it's frustrating when stuff takes a while to come back. I'm not going to promise them for Wednesday, but I will try my best to get it for you for our next class. Otherwise, definitely by a week from today. So, but hopefully by Wednesday, without actually promising. It's like the half promise. Like, you know, when you see someone and you're like, oh yeah, we should hang out sometime. And you're like, yeah, we should hang out sometime. And you're like, yeah, cool. Anyway, I'll be better than that. All right, now that you've taken your quiz, let's watch a video. We talked a little bit. Question, Carissa, go ahead. Oh, they took the quiz. Oh, Will, would you mind popping the door there and summoning anybody that's outside and letting them know they can come in now? Thank you. Thank you for telling me that. I didn't realize they were waiting outside. Tarika, yes. Is there anyone out there? So there's a couple of people taking the exam at the testing center, and so I asked them not to be here because then they'll see the questions in advance to explain. Is there anyone out there? All right. Thank you so much, Carissa. Oh, good. Okay, come on in now. Perfect timing, Alan. Oh, you could come in too. You already took it, so you're good. But that's fine. You're here. Good timing. All right. So just a quick transition here. We talked about how newts in the newts, which are salamanders in the family Salamandridae, have these toxins on their skin. With the example of the I can't think of the common name, Notothamnus viridescens, the newt that lives here in Ohio. What's the common name? The Eastern newt. Thank you. Yes, I don't know, but I can remember Notothamnus viridescens, but I can't remember Eastern newt. So it includes that species as part of this group. I'm going to show you a video here about another species in this group in the genus Pterica. And this is just a short video kind of to transition from quiz brain to herpetology brain. Not that those should be two different things necessarily, but. And bear with me for a second. I may need to adjust the sound. I do. So I'll start it over once I get the sound set. I didn't record it, but I'll record it this time. So aposematic coloration is one term I want to make sure you take away from there. And that is essentially warning colors. And in the video, you saw the newt arch his back and display this red or orange underbelly. It's basically the opposite of camouflage. So camouflage is when you try to blend in with your surroundings so a potential predator can't see you or detect you. Aposematic coloration is when you like say, hey, I'm here, but if you try to get me, that's going to be your problem, right? And so that's why we have the red F stage of the eastern newt. It has this bright red colors. It's like a warning signal to say, like, you see me and you can eat me, but if you do, it's going to be trouble for you. And we see this in a lot of examples of a lot of examples in amphibians. Another famous example is like a poison dart frog. You see these crazy frogs from the tropics that are like blue and yellow and green, like bright colors, like neon colors almost. And that is aposematic coloration. That's a warning signal, and it keeps them safe because if a predator can say, okay, I see that frog, but I ain't gonna eat it, that keeps the frog to hopping around for another day. I saw Lennon and then Mason. Go ahead, Lennon.
I was just gonna ask, you mean stuff like the poison dart frog?
Yeah, exactly. I channeled your energy, and then I answered a question for you. Yes, Mason, go ahead.
Do we know like how that coloration evolved in those species?
How it evolved? That's a great question. So where, I don't know. People have studied this in detail. In general, the idea would be that some kind of random mutation changed the color of the skin, and that proved to be beneficial. Or it could be that the skin changed color slightly for a different reason, and then over time that was kind of co-opted because it was recognized by potential predators. So I don't know the exact details in like these guys, but in general, it's just like this kind of chance coincidence that the toxic animal, you know, some of them were a different color, and that allowed predators to identify them, which kept them safe, and so then they passed on their genes more. Yeah, that's a great question, actually. I'd like to know more about the details of that. And my guess is that it's going to be different in every clade that's evolved this kind of aposematic signaling. So, good, great question. Chase, go ahead.
For this newt, or like I guess any other animal that has like that coloration that's like associated with toxicity, does every time it like use their toxicity like trait, like this some kind of bacteria in its skin, does it need to like regenerate some, or is it just like always there?
The toxic, the chemicals?
Yeah.
Yeah, that's a great question. And so, yeah, it can use up and needs to generate more, and it varies among individuals. It can even vary on parts of their body where there's more or less of the tetrodotoxin. I couldn't tell you exactly what other factors affect that, but it's definitely not a kind of always there constant thing. And the frogs are actually a great example, is they derive the basis for their toxins. They're not neurotoxic like these guys. It's a different kind of toxin, but they require chemicals in specific kinds of prey, kinds of ants that they eat, and they process those chemicals to produce this toxin. If you keep poison dart frogs in captivity and feed them crickets or whatever, they can't make that toxin, so they're actually not toxic. So in some cases, they need to acquire that chemical directly from their food source. In other cases, they have these bacteria, symbiotic bacteria that produce the toxin. It's cool stuff. It's the same, actually, the toxin in these guys is the same in pufferfish. Like in the sushi that you can eat that's made from pufferfish, where if they prepare it wrong, you can die. Do you know about, yeah, don't eat this. I think it's a terrible idea. But yeah, it's the exact same toxin. It's a neurotoxin. Cool. All right, I just wanted to show that and bring some concepts in, specifically aposematic coloration. That'll come up again, and just give us a little break after the quiz. Before I get into our first activity here, though, I do want to fix a mistake. Princeton very kindly pointed out that last week I incorrectly said that amphisbaenians were commonly called slow worms. They are not called slow worms. Slow worm is a common name for the lizard Anguius fragilis, which is a legless lizard native to Europe. Looks like, oops, that looks like, oh, where'd my picture go? Hold on one second. Oh, weird. All my graphics for my PowerPoint are jacked. Give me 1 second here. Nope. You seeing that okay? Yep. Okay. So it looks like this. Essentially looks very, very snake-like. However, there's some characteristics that allow us to know it's not a snake. For example, it has external ear openings and has a very, very short tail. And we'll talk more about that when we get into squamates in a little more detail. I have to share a picture of my daughter holding one of these guys we caught in Croatia a couple years ago. I was super excited, like I lost my marbles. We found this legless lizard off this trail that we're at, and it's a cute picture of her, so I wanted to share that with you. To be clear, amphisbaenians are sometimes called worm lizards. They are neither worms nor lizards, but that's where I got my terms mixed up between a slow worm and a worm lizard. So I just wanted to be clear. I appreciated that, and if I ever do that again in class and you notice something, don't hesitate to let me know. I will be grateful and not feel put out at all. Cool? Awesome. I'll tell Anna that I shared a picture with her in class. She'll be happy to know that. All right, so for today, we listened to a podcast, a long interview with Priya Nanjappa, who you see here. And what we really want to do today is debrief it a little bit. And I want to do this in small groups rather than our big groups here. And so what I'm going to do, instead of assigning groups and having you move around, we're just going to do it at your table. And I guess I should be more clear, like half a table. So like 4 people that are together at the half a table are a group. And what I'd like you to do first is spend 5 minutes just discussing this question. What did you think of the podcast overall? Did you learn about amphibian biology? Did you learn about being a researcher? Was it enjoyable? What did you take away from the experience? So just 5 minutes in your small group of 4 people. Or 5, no, all of them are 4. We'll bounce around and then we'll move to the next round of discussion. Sound okay? We good? Okay, let's do it. I'm gonna pause. That's too bad, okay. Yeah, okay, yeah. Well, let's pretend you have the opportunity. Oh, yeah. Wow, it's a little bit disconcerting that the fake laughter was louder than the. Okay. Anyway, you can spend one night in a place where these guys are mating, and I want to know where they're breeding in large numbers, so like a whole group of them get together, and we'll be able to see some of this later this spring. If we take some evening field trips in March when the weather changes a little bit, which is really cool. But what I want to know is, what question do you want to investigate? What do you want to take the time to try to learn, and specifically, what aspect of toad reproduction do you want to learn more about? It could be because you're interested in it. It could be because science as a whole doesn't know the answer to that. And then thinking about the question you want to ask, I want to know what kind of data you're going to collect to answer that. We focused on the mud puppy paper you read last week, for example, on thinking about what data were collected to answer their questions. Now, I want you to think about what data you would collect. For the purposes of this discussion, don't worry about technology or logistical implications. Let's say you really want to measure the size of the testes of the males or something. Don't worry about the fact that that's logistically challenging and you need to kill the toad to do it. Just think about your brainstorming here, right? We're not killing any toads in this process. And then think about how those data are going to help you answer that question. So a little more detail here. You can do it how you like it as your table. You can kind of come up with one experiment you all want to do. Each of you can share your own and bounce around ideas or however you want to do it. Does that sound okay? All right. I would try to come up with another good frog pun that I can throw in there, but I'm going to stop. Yeah, go for it. A little bit as the full group here. I heard some great conversations at some of the tables. I apologize I wasn't able to get around to all of them in that time. The key takeaway here is learning about amphibian reproduction, and then thinking about how you test ideas you have, how you test the hypothesis, and what data you need to test the hypothesis. A couple of themes came up, but I want to just give you some vocabulary and make sure you've got these terms down. The first one you see over here is the term reproductive skew. This is the idea that within a population that I'm just gonna talk about male reproductive skew right now because it fits with amphibians, but it could be female as well. Is that not every male contributes to the next generation the same? So, for example, let's say we have a pond where there's 100 male and 100 female American toads breeding. Just to go with the simple example for what I do. In that pond, unless there's some, physiological problem with the female, all the females are going to produce a bunch of eggs, right? So all 100 females produce eggs. But not all of the males will fertilize those eggs evenly. Maybe 10% of the males will father 70% of the offspring or something like that. And so that is that concept that not all of the individuals in a population are contributing to the next generation genetically the same is called reproductive skew. And that ends up being really important to understand mating systems and how genes are transmitted to the subsequent generations, and therefore how evolution and selection could work. Not necessarily selection, but how evolution through selection on those genes in subsequent generations might work, and who is fathering or parenting, to say it more generally, the offspring of the next generation. So that's the reproductive skew, is the idea that they're different individuals contribute different amounts to the next generation. We just published this paper on this idea in garter snakes earlier this year, or last year, I guess it's 2022 now. The idea being that in garter snakes, we have something called multiple paternity. So let's say that a female garter snake is producing, they give live birth, right? These guys do. And so let's say she gives birth to 12 babies. Within those 12 babies, there could be, they could have 2 or 3 or even 4 different dads. And so we did a study using genetic analysis to get all of the babies, and we knew the genes of all of the moms, so we could infer how many dads contributed to each litter, and we could quantify multiple paternity. So how many of those lizards, those litters had genetic contributions from multiple males, and then also how skewed that was in the population as a whole. Were just certain males contributing much more genes to the subsequent generations? So that's a cool concept. I want to make sure you got that. The second concept I want to make sure you got is intrasexual competition. Intra, of course, meaning within. So this is competition within a sex. And the most common way we see this in the example that we're talking about right now with the toads is males competing for females. And the way a male toad tells a female toad that he is awesome is by his call, right? And you heard the American toad call. You heard in the interview she was imitating it. Princeton could do a good imitation of this one. I won't put him on the spot though. But this idea that males need to compete to see who can attract the female to them. And the females can assess this by the male's calls, and they can tell certain things about a male based on that call. And specifically in frogs and inurans generally, the duration of that call is really important. So how long and how loud a male can call. How loud he can call affects how far away a female can hear him and therefore find him. And how long he can call kind of shows off his endurance. And a female is going to want to select to produce offspring with a male who has great endurance, is generally more physically fit, and is therefore more likely to produce and contribute to fit offspring. So intrasexual competition. This can take a bunch of different forms. For example, in rattlesnakes, they fight, and it's super awesome. They bring their heads and their necks up, and they try to push each other to the ground. They never, ever bite. but they do like this awesome wrestling dance thing that's like super cool. And there's tons and tons of examples of that intrasexual competition. There's also intrasexual competition with females as well, but it's usually not as in general in reptiles and amphibians mating system. It's more common in males. Amplexus is a term that we brought up earlier when I talked about amphibian diversity. I showed you some videos. It's basically the act of intercourse without actually having intercourse. So it's external fertilization. It's what frogs and toads do, with very few exceptions. The female releases the eggs. The male is in very close, if not on top of her, and he releases sperm at the same time. The sperm meets the egg outside of the female's body, and that is external fertilization via amplexus. And that is in contrast to fertilization via an intramittent organ. And we talked about that a little bit with hemipenes in squamates, and we also talked about that a little bit with the phyllodeum of the Sicilians. So just some concepts that came out of that. I want to make sure you know those terms. We've got aposematic coloration from here earlier today too. Let me just pause my talking and see what questions you have about that stuff. Mating systems are neat. I say we put that in your notes. Mating systems are neat. All right, well, let's, what time? Let's take 5 and come back for the next part of our class. We've been cranking for a while here. Looks like let's come back at 10 after on that clock up there. And I'll have things ready for our next fun activity in herpetology today. Sound all right? Great. All right, let's bring it back here. I see a couple people outside. We'll give them a quick second to come in. So what I want to do now is, we're originally going to do this last Wednesday, but I didn't want to do this with an online class, which seems a little odd to not want to watch a video with an online class. Here's my logic for this, is that if we watch this video together, one, we're going to be focused on it. I know if someone gave me an assignment to watch an hour-long video on YouTube, I would kind of be half watching it while I probably did some other stuff. I don't want to speak for everybody, but it's difficult to concentrate. Also, there's lots of places here where I'm going to pause it and talk about concepts and see what you have questions or whatever. And I think it's important to be able to do that in person. We won't finish the video today. We'll have to finish it on Wednesday, but that's cool. I did put a link. This entire episode is available for free on YouTube. I put a link to it in your program for Wednesday as well, in case you're not here right now and you're watching this video, or you want to go back and review anything. In conjunction with this, I've got a couple questions I'm going to ask you to reflect on as you're watching, and then these will be due in Blackboard by next week, Monday. So you have a week to do it, and it's worth 6 class points. And the questions are, and I want to give you the questions now so you can think about them as we're watching. Generally speaking, what adaptations allowed vertebrates to colonize land? And you'll hear about that in the video. But remember that our ancestors were all completely aquatic, and then the first terrestrial vertebrates were amphibians, or the transition between fish and amphibians. And so what allowed them to do that? What's something you learned about amphibians that was new or surprising from the video? And describe one aspect of amphibian biology that we should study more, you think is worthy of further study. And the final question is, on a scale of 1 to 10, how freaking cool is the giant salamander? You'll see I gave you a little bit of help with this question. It's a multiple choice question. It's got 10 possible answers, and I gave you a little bit of aid with this one. So hopefully you'll get that one right when you answer it. So that's the scoop. We're going to watch the video now until close to the end of class, and then I'll just wrap some things up with some announcements. Any questions about these questions or the expectations? Yeah, Alan, please.
So is this assignment going to be open Wednesday after we finish the video, or is it going to be open?
Thanks for asking that. The assignment is open, will be open after class today in Blackboard. And I did it in Blackboard because it's a little bit longer answers than like a survey, and I thought you'd probably want to type it instead of thumbing it on your phone or whatever. So it is available, if not right now, I think I set it to be open by the end of class today. Mm-hmm. Yep, please, Sam. Regardless of when we finish the video, it's not due till Monday before class. So you'll have a week to do it anyway. I would recommend not waiting because then you'll forget what you're thinking about, or at least while you're, I want to tell you the questions now, so while you're watching, you can take some notes and then put your answers together. I wish we could have snacks in this room. I totally would have brought popcorn or something for us, but alas. Okay, let me just transition here and get the video on the big screen. This will just take a moment. Princeton, would you mind turning the cam lights all the way down? I think we can do that here. It's like dark. It's like being in a movie theater. Like that was like the trailer. And now, why isn't there sexual size dimorphism in all animals? Or why are females bigger in all? Yeah. That's a great question. So Chase's question is why don't we see the same pattern across all different organisms? And the reason is because different organisms have different reproductive modes and different ways of finding mates. And so in some cases, females aren't limited by body size. So you can think of, I'm trying to think of a good example. A Knowles is a kind of lizard, and they always, most of the species always produce one egg at a time, right? So in that case, it doesn't really make any difference for the female. She's not trying to pump out thousands of eggs at the same time like one of these frogs. And at the same token, in some cases, it might be beneficial for the males to be bigger if there's things like male-male combat for mates or things like that. And we see lots of examples of that. So it just depends on how they find mates and then also how they are actually producing eggs or sperm and how that might be benefited by being larger. That's a really good question. Yeah, Ethan, go ahead.
Could it also be a case that you don't have any like toxins or protect yourself, but if you do get larger, you have to attack you.
Yep, there's trade-offs there for sure. You can become a target of predation if you're larger. You might not have access to certain like burrows or things like that if you're bigger. So there's definitely a downside to being too big too. Good. So sexual size dimorphism. I want to make sure you got that. Anything else? Anybody else want to jump in? All right, great. Let's wrap up this section on the. Okay, so we'll finish that up on Wednesday. Just want to give you a quick reminder that our mud puppy trip will be next week. We'll go Tuesday night and Wednesday during the day. I'll put out a survey to see who's going on what trip. Remember that class requirement is to go on one of the two. You're welcome to go on both if you want. You know the details. We've talked about this a little bit. So just a reminder to get that on your calendar. Also, oh, wait. Oh, just kidding. Is that all the reminders I have? And one other reminder for you. Oh no, I didn't. Actually, that was it. So that's all I got. Thank you, everyone, and I will see you on Wednesday. I'll stick around if you have any questions.